great to have you in person. I want to start with your background, right? You've got a pretty unique one for the space. You worked in healthcare investment banking at Goldman for 2 years, right out of college. You studied neuroscience at Vanderbilt, but now you're the CEO of a biotech startup. So you're obviously quite knee-deep in the science. Talk to me about that journey. Like, what made you want to decide to step out of a more conventional finance or even a more conventional operating path and like do biotech? And yeah, why science? Why biotech?
Well, thank you for having me. And the short answer is I love science. I'm a total nerd deep down. I worked in labs in high school and then in college. I worked in particle physics labs and then chemistry labs, and then really got excited about neuroscience in the same sort of vein as it being like a fundamental question that you get to ask, but also having that direct translatability into medicine. So that was really my first exposure into medicine, and then seeing this ability to, you know, do science, but do it in a way where the translation is really meaningful, impactful, directly helping people. It was awesome. So I was in this great lab at Vanderbilt that was built out by teams from pharma that then started a lab. So they had like 50, 100 people there working
Wow.
on a full drug discovery operation for neuroscience, for neuropsychiatric diseases, schizophrenia, all
Mm,
those. So when I was leaving undergrad, I really wanted to learn more about the business of science, thinking that maybe there are ways to sort of maximize the impact of science. I worked in banking, which gave some good exposure to all of that. But at the same time, we were just talking about how um, you can look at those companies and it's just such massive size and capital needs. And back in 2010, 2012, there wasn't as much of a biotech VC community.
hmm.
So I
Interesting.
left banking being like, well, you can't start a biotech company and I want to do something entrepreneurial. So I moved into the world of tech
Got it.
and worked in tech investing, and that got me a lot of exposure to entrepreneurship. But really, in the tech world at the time, it's just so much business model innovation. So, what I found interesting is that there's obviously a lot of innovation in science, but not a lot of business model innovation. On the tech side, back in 2012 to 2016 timeframe, there wasn't AI. There wasn't a lot of totally brand new technologies, but there was a lot to be done with software, enterprise software, data. So a lot of business model innovation. And, to keep it short, essentially, Syenex, the work I'm doing now, is all about combining those two things, taking, the best of science innovation, the best of business model innovation, trying to find a niche where there's major, unmet need for impact.
Yeah, totally. Total side note here. Where was Flagship and Atlas back then in 2012?
I, I don't really know. I mean, they did have some big companies. I think off the top of my head, I think there was some early microbiome businesses being started.
Mm,
I think Moderna had just been launched around then,
got it.
but those were massive
Yeah.
projects. It wasn't, I guess you could say founder-led biotech was still super early.
Yeah. I mean, a lot of people say that it's still super nascent,
It
right?
is. I mean, not to do too much of a Science plug, but I think this opportunity over time is that, and Nucleate does this a lot as well, right? If you make it easier for people to start companies and you make it less capital intensive to go from an idea to clinical validation or impact, then you can imagine more founder scientists starting companies
Yeah.
and more venture capitalists being able to come in because it's a little bit more accessible and you don't need these massive syndicates just to even get into that initial proof point, which is what, pharma and the ultimate buyers of medicines are really looking at.
Yeah, that makes sense. I feel like it can be very intimidating to start a biotech startup. I don't know if we're in a biotech winter still, right? There has been a surge of VC funding, even just last quarter, for smaller startups, smaller biotechs. So I'm feeling optimistic about that, but maybe not as optimistic about like, just how hard it is and how challenging it is. And you can definitely speak to some of those challenges, right? As a CEO, as a founder. Talk to me about that. Like, what were some of the early challenges starting Syenex, and were there any points in starting this business where you felt like, ah, like, this might not get to where I want it to be?
It's a great question. One thing to what you were saying, the thing that gives me a lot of hope for the field is the fact that there's so many people that have committed their careers in a way
Yeah.
to working in drug development, biology, synthetic biology, bioengineering. And that doesn't change overnight. The market cycles as it goes, but there are a lot of people that want to make impact. They want to do it through medicine. They want to use synthetic biology and all these great innovations that are, secularly moving the field forward. So, there are always going to be constraints on the capital side, but that will ebb and flow. So that's what we're in. But that then relates to the hard parts on the company formation side and building, especially a company that's effectively like playing a derivative to the core drug development work, meaning we are supporting drug developers. If there aren't venture capitalists supporting drug developers, then it's hard to then build a business that supports drug developers. So I would say the hardest thing with Syenex at times has just been the question of like, are we too early, right?
Mm.
And too early is wrong, right? So you need to always be
I think it's
thinking
better than
about that,
too late, probably.
probably. But um, you know, there are many ways to be wrong. So
Yeah. No, that makes sense. I want to talk about Syenex more in detail. It's spun out of Josh Leonard's lab at
mhm.
Northwestern, correct? The specific breakthrough there was that we can genetically reprogram producer cells so that the cargo can be selectively packaged into these things called extracellular vesicles, rather than loading cargo into a vesicle after it's being made. Talk to me about this discovery. What made you interested in engaging with this science? What made you think like this patent, this idea, that's what I want to be in on, and that's what I want to start a company around?
Yeah. So I've been working in venture capital for a while, focused on synthetic biology, cell and gene therapy, and I spent a year or so as a visitor in the church's lab, and it was when I was there that I got to really see the power of applying synthetic biology to drug discovery, sort of multiplexing, sort of high throughput ways of leveraging the, atomic factory of a cell with DNA. So everything around DNA synthesis and sequencing is like a critical uh, tool to ultimately do really complex work and discovery multiplex and high throughput. And Therapeutics was being formed at the time, and it was just a great example of, I think now they call it, multiplex libraries or and machine learning, the ML, and I wanted to do the same thing in extracellular vesicles and in vesicle engineering. In the, I want to say like 2018 timeframe, there was a lot of talk about exosomes in general, more
Yeah.
in the form of like, when this cell produces an exosome, it goes to this place. When that cell produces an exosome, it goes to a different place. They have these natural tropisms, and thinking about what could be driving that related to sort of the proteins on the surface of those particles. So um, finding Josh and Josh's lab was all really a search of, are there tools to use synthetic biology to genetically encode the proteins on the surface of those particles so that you could really interrogate the driving mechanisms of these delivery particles? Because delivery is just such a massive problem in genetic medicines, holistically, cell and gene therapy. So that was really the idea, was saying, what would be the best way to solve the delivery problem? Looking at the field of exosomes as sort of an early sense of unique tropism that's maybe natural, maybe not, we don't know. Still very early science. And then finding someone that had been spending so much time developing these really powerful tools to design exosomes and to interrogate delivery. And then I would say the big thing with Josh was really getting to know him. We chatted for a long time, years, before starting the company, just to make sure we really
Oh, cool.
knew each other. We're on the same page. We're philosophically aligned with the model, because um, when you start a biotech company that is taking a platform business model, probably for at least the first 2 years, everyone would say you should make drugs. You should become a drug company. Don't be a platform company.
Yeah.
You're making a huge mistake. You can get rich fast if you
Yeah.
made drugs. Um, and we were dogmatic, not about technology, but about that model.
Interesting.
And um, and we still are. There's still people that'll say that, and it could be totally wrong, right? That's a big risk, but the driving force is, or a North Star, if you will, is impact. And that may sound cheesy, but the idea is that if we can make great technologies that people need and you make it broadly available, good things will come, and we'll find a way to get a piece of that, keep ourselves afloat. So, yeah. So it's all about the delivery problem and exosomes as a great medium for solving the delivery problem. And there are some interesting insights along the way, really around viruses being, in effect, one of the earliest examples of exosome engineering, envelope viruses. So there's a lot you can learn from envelope viruses when it comes to efficient ways to do exosome engineering.
Cool. Yeah, I want to talk about the delivery problem, right? I mean, let's just start with the basics. There's the clear lipid nanoparticles have been established for a while, and then viral vectors, AAVs, can carry relatively smaller cargos, right? But also not as great for specific delivery. Their tropism isn't great. And also there's lentiviruses, right? And retroviruses and different types of viruses. So can you maybe explain, like, what is an extracellular vesicle? What is an exosome? How is it different, and why do you think that it's the best solution for delivery? Because people are tackling this problem through a lot of different ways, right? Like, even I think for the viral space, like pseudotyping is something that's being done to try and tackle at that. So maybe you can explain the fundamental of the science.
Yeah, the taxonomy of delivery, you've got biologics, you've got synthetics, you've got some hybrids.
Yeah.
On the synthetic side, you've got liposomes, lipid nanoparticles, polymers. There are lots of things. On the biologic side, you can be really, I feel like there are two big categories. They're protein capsids, and then they're vesicles or envelope systems, right? Something that's either blebbing off of the cell with membrane or something that's being excreted and it's sort of a closed protein. Those protein shells, that's like adenovirus, AAV, they work great. They have a lot of amazing qualities. The tropism is challenging only because the nature of holding the DNA, the genome, inside this particle and having it all come together, I always envision it like protein origami that just needs to be perfect,
Mm.
easy to break, you know? Vesicles have their own sort of protein shell inside, and that needs to be, carefully managed and not broken.
Mm.
But the tropism can be driven by proteins decorating the surface, and that can happen through genetic expression in the cell. That's sort of the factory making the particle. And that is much more robust in a way from a design standpoint. You can put almost any antibody on the surface and any sort of natural protein on the surface, or even, synthetic protein on the surface. So it's for that reason that I got really compelled by this field, specifically because there's it's such a large solution space that you can operate in without breaking it. And then inside the vesicle, there's some really amazing science that's been done by other labs that shows it's possible to um, effectively further re-engineer the nucleocapsid of a lenti or an envelope virus, so you can use it to chaperone other proteins. And so it could be ribonuclear protein complexes like Cas9, base editors, prime editors. You can do that alongside genomes of, viral-based genomes, collectively allowing for targeted gene integration. So there's just a lot of interesting things you can do with this. And because it is a DNA-defined system, meaning DNA goes into a cell reads the DNA, produces the particle, you can kind of boil it down to a DNA problem, DNA design problem. And everything in DNA design is improving, insanely quickly, right? Whether it be the cost of synthesis, the cost of sequencing, or even better, designing proteins, right? And being able to then translate that onto DNA. So all the AI protein design companies, that becomes an amazing tailwind. So there's just so much that you can do when you're in the biology space, when you're in the DNA design space. So that's why I really like it. Now, you know, I'm open-minded to other delivery systems. I would say the dogma is all about the business model, not the fundamental technology. And, there are these hybrid particles that are actually pretty interesting,
Okay.
where you're combining protein binders with lipid nanoparticles. So targeted LNPs is a big category, where you conjugate up an antibody to an LNP, and now it's much more targeted, much more specific. So we explore that pretty deeply. There are cases when that's going to be a great delivery system for certain types of payloads, especially when you have chemically modified nucleic acids like antisense oligonucleotides, chemically modified RNA. But if you want to deliver something that a cell can produce, these vesicles work pretty well.
Talk to me about like your dream payload.
Dream payload?
Yeah.
Well, there is targeted gene integration that opens up a lot of doors, especially for, to answer that question, I have to really think, what will drug developers want to be able to do and what sort of problems can't be solved today? For payload, epigenetic editors
Hmm.
is pretty interesting. There are a lot of things people have done on top of sort of that base Cas9 framework for that. And then targeted gene integration. Those two I find, and you can even
They're
combine.
fascinating. Yeah.
Yeah. I mean, we all, all of our cells, for the most part, have the same genome, right?
Yeah.
So the difference between your retina and your skin, is epigenetic mostly. So being able to modify that for diseases of aging, um, for all sorts of indications can be pretty interesting.
I want to talk about Syenex's business model. We've been talking about how Syenex is a platform company, right? And talking about Moderna is a really great example of a successful platform company. But they did, eventually pivot to eat an entire disease space. And their logic, right, is that the tech is really valuable, but where the exponential returns actually come from is owning that commercial drug. And so Syenex is committed to a pure partnership model. Maybe you can talk a bit about that, maybe a bit of the rationale behind that, and do you ever see that changing?
I can talk about this for hours. So
Yeah.
on the platform side, the platform business model, I think about this all the time.
Yeah, yeah.
We were saying earlier, platform is used to describe technology and is used to describe a business model. And I think a lot of times it's used sort of without being clear which one
Mm,
is being discussed. From a technology standpoint, platform technologies are amazing, right? By definition, what it basically means is a technology that can be used to enable hundreds and hundreds of drugs, right? That's a real platform.
yeah.
Um, Now, the problem, in my opinion, is when a platform technology is in a drug company,
Mhm.
it's not, it doesn't get to realize its full potential. And my reason for saying that is because of just sort of the natural history of drug companies. And frankly, the cost of developing a drug, bringing it into the clinic, bringing it all the way to commercial, launching it, that's a huge, huge expense. I mean, people say huge numbers, $1 billion, whatever it is. And it's very hard for one company to be able to pursue more than two drugs, right, in earnest. So you have a platform that could be enabling hundreds of drugs. What are you going to do? Moderna had a lot of really creative partnership structures at various stages of its arc, um, but was ultimately built as a drug company. Maybe with some interesting ways to increase the number of drugs in its effective pipeline. The platform business model, at least one that you see in the field of tech and software, is one of the great things that you'll hear about a platform is a platform is only as valuable as the amount of value built on top of it. And how do you build a platform that allows drug developers to get a lot of value out of it? And the way that we've been doing it differently, especially in the delivery space for genetic medicines, has been build amazing delivery solutions that can enable as many drug developers as possible and license it to them non-exclusively. Essentially asking the question of like, what would they wish existed? And try to build that. And what they want is a royalty-free delivery system, right? That solves a problem, prevents it from, today drug developers have to either build it themselves or wait for things to go off patent 20 years later.
Yeah.
Because essentially everyone else that has delivery systems is a drug company. And drug companies need to really focus on their drugs. So if they're ever going to partner, it needs to be really worth their time. That opportunity cost is so big. They've got huge budgets going into their lead drugs. So to distract that team away from that is costly. For us, we don't have a pipeline, so we can focus only on the platform technology and the platform business model, right? We spend money on R&D, preclinical, design R&D, marketing, and legal IP.
Yeah.
and licensing. We license it all out. So that's the business model, and it's just been really exciting to see it on the T cell side, because that's where we've been starting. T cells are like half of the genetic medicine market. It's a super impactful cell type, long lived. I mean, there's studies that show like T cells going from one mouse to another mouse to another mouse, like generationally. They live for a very long time. So T cells have huge implications, or can be used to treat cancer, autoimmune disease, fibrosis, and that's just the beginning, right? That's just like making it do what it normally does and target something, versus using it as like a little submarine in your body that you can engineer to do anything. Um, but when it comes to in vivo delivery to T cells, which has really been our like flagship product right now, VivoCell, uh, it's just been really amazing to see all the diverse programs, if you'll call them that, or engineering strategies that drug developers have for those T cells. If you look at the world of in vivo CAR-T, where the drug developer is building the technology themselves, the delivery technology themselves, for very valid reasons, they focus on CD19 CAR, right? It's
Yeah.
the most proven, sort of the safest. You don't take biology risk on top of technology risk. And that makes sense if you're doing it all yourself. But when we are taking technology risk and able to invest in that, it unlocks biology risk for others that are using that technology. So we're able to, I think there are a lot of economic theories when it comes to collaboration, but like when you have two different groups specializing on two different things, the collective is that much better. So that's how we see these drugs that are able to be built on top of the work that we're doing. We're solving the delivery problem, but what we really want to do is abstract away as many layers as possible, such that the final thing is the program that the drug developer wants to integrate into a cell. So in a way, it's like we're building an operating system for genetic medicines. That's been an idea I've been toying around with, just as a way to better envision the opportunity and the scale of what is possible here. It's almost like these different delivery programs or delivery systems are like, vector developer kits that they can then build on top of. It's almost like building individual applications on top of a broader software layer. But I think in general, more abstraction in biotech is the path to reducing the cost of drug development, and reducing the cost of drug development is the path to more drugs and more cures.
I'm really curious. So I just think platform companies are beautiful. For many of the reasons you've already talked about, just this potential, this unlimited potential to cure thousands of diseases with whatever you're building. And that kind of inspired me and made me want to go into the diagnostic space, at least in undergrad, because I see that modularity, the applicability, like being nimble. I really enjoy that. But it's also incredible challenge, right? To do partnerships, and each client is different and requires different things. Each relationship is different. I don't know how many conversations you've had um, so far. I assume many, but maybe you can talk to me a little bit about that, how you adapt
I will
or
give
thinking
I will
about it.
give credit to Eric Anderson on this one, but when you're in the venture capital world and you're meeting with companies, it's thrilling. You get to talk to everyone. You hear all these great ideas. The only problem is you have to say no to everybody. So you get to hear it all. You get to vibe with them. You talk about it, and then you're like, oh, actually, I can only say yes to 10 over the next 4 years, so no to you. But how can I help you, right? And so many conversations with VCs, and how can I help you? Because I can't give you the money that only a few people can get, because I promised my investors I'll only do 10 deals or whatever it is. Um, so now being on the platform side, it's so great because it's like, how can I help you? You skip that whole investment part, and it's just you know we raise money, we sort of invest our profits, we make technologies. Hypothetical, cost us $10 million to make the technology. Maybe it would cost someone else 12 million because we've got the expertise, or 20 million or more. And we can license it to them for, 1/50th of that. And we're like, well, we're going to license it to, at least 100 companies. So we'll be fine. So we're helping you. So it's a way to basically have that same sort of VC deal flow, VC sort of outreach, and meeting with tons of
Mhm.
great, smart people, and just having a much higher hit rate of helping them. So that's been a lot of fun. And the platform side, it's funny that you said that, thinking about it in high school, it's not uncommon. I think most bioengineers, I'll say, scientists that think about biology as something to be engineered, that really sort of have a natural inclination to platform technologies, um, imagine a platform business model as the right way to bring it to market. And I saw that in the church lab, tons of super smart people, and orbiting around it in other labs as well. Lots of people make these amazing technologies, and they envision like this could be so impactful because I can imagine 100 drugs that could be built on this platform. And I also have seen many of those scientists turned entrepreneurs, be very strongly advised that the right business model is making drugs. And, the advice that I haven't seen pan out great is like, do a proof of concept drug,
Mhm.
bring it into the market,
Just
basically.
show that you work.
Yeah,
Yeah.
but that just never happens because you sort of, you know, it's like trying to live a proof of concept life. You don't get round two, and that's what it feels like. They do a proof of concept drug, and next thing you know, that's the drug. That's it. That's the company, and there is no round two. And worse than no round two, they may even have a better drug idea, but after you're in the clinic, you know, after you're in the clinic and you're a biotech, every new idea, new drug is more of a liability than an asset.
Hmm.
It's so expensive to raise that drug, than it is to just say, let's just stop making drugs. Let's focus on advancing our drug. So I just think there are a lot of really smart people out there that have really great ideas for platforms, and they haven't found the right home for them or the right path for them. So, um, the big derivative success for Syenex would be just creating that precedent
Yeah,
for others.
totally. Is there something to be said about platform companies de-risking a little bit? Like, never being able to be tied down to, like, one particular disease or indication? And I imagine that that might translate to something that could be economically smart to do. Is that something that you have considered or thought about?
Yes. Yes, and a lot. No, it's so,
Yeah.
no, it's a great question. I mean, like, I've wrestled down that same sort of a similar sort of thinking, which is like, if it wasn't a platform, what indications, what sort of drugs would I want to work on?
Yeah.
And just like anything else in entrepreneurship, it just has to be something you're deeply, deeply passionate about, right? The
Yeah.
thing that you keep working on when everyone says you're wrong, when it is a bad idea, not could be, but just straight up is a bad idea, and everything's wrong, then you still say, I'm going to keep going. So that you got to find that. And um, if you don't have that, yeah, it's hard to start a drug company. But there are a lot of people that feel that way. And, you know, when you realize that's what they're feeling too, it just makes it that much more meaningful to find ways to help them. Um, but yeah, when you're working on a platform technology, you obviously aren't tied down to one indication or one drug. You're able to empower many. You sort of have a different problem set. I like to call it merchandising, just because you have to think about what are all the different things people may want to do. What are the problems that you'll need to solve? You have to talk to, I mean, do we talk to people? All I do is talk to people and, surveying the land and trying to get a sense of what's the Venn diagram, what's the big overlap so we can focus on that. And then what's the next biggest thing and focus on that and just knock them out. But there are some complexities with the platform model. And I would say that there is this risk frontier that you need to be mindful of, and I've seen this. If I've seen people try to launch platform companies where, um, the technology that they're passionate about is beyond the risk frontier, is beyond the comfort level of the counterparty that would be using it. So, yeah, when it comes to making platform technologies, you need to be really mindful of this risk frontier. It's obviously not easy to find. You need to sort of talk to people to understand it. But the nature is there are certain technologies that a drug developer that could be a potential partner won't feel comfortable adopting unless it's been proven in the clinic. In which case, you see those companies sometimes say, okay, great. Listen, heard, I'm going to do a proof of concept drug and show it works in the clinic. But in the sort of decision tree of how to build a platform company, the first thing is never make a proof of concept drug. So
Hmm.
you got to be wary of that. So you do need to be mindful of the risk. And if you find yourself there, you need to dial it back. When we started, we wanted to do in vivo delivery to any cell, right? That was like mission number one. And when we talked to drug developers, at first they said, we're not ready for in vivo T cell engineering, but we'd love better ex vivo. So we launched some products in the ex vivo engineering field. And it was great because the T cell biology is the same outside the body, inside the body, or at least there's similarities. So there are a lot of critical learnings that we now apply to the in vivo work. And it allowed us to, build up the team, build up the platform, build up discovery. But it was just an example of where that risk frontier was and how it shifted over time.
Maybe you can explain for the audience that ex vivo versus in vivo problem for CAR-T therapies and specifically what you guys are trying to do with your partnership with Lucid Bio.
Yeah, absolutely. So T cell engineering, as I always, will describe, is like, take white blood cells out of the body, you reprogram them to see the cancer, and then it kills cancer. And it's worked very well. And it's pretty amazing. Um, the history has been what I said. You take the white blood cells out of your body, leukapheresis, and then you engineer them ex vivo outside of the body. There are a lot of different ways to do this. Most people use an enveloped vector, lentivirus, gamma retrovirus. There are also really great mechanical poration systems. Portal Bio has a really powerful one. Some people use electroporation. There's Maxite that's really good for that. And they engineer them in various different ways, integrating a gene, editing a gene, things of that nature. But to do that, it requires kind of a logistics nightmare, right? You need to take a patient who is in a rush to get treatment, right? Take blood out of them, freeze it down, ship it across the country, send it to a lab, have it be processed by like 10 people with all these different, non-automated systems as of today. Put it back in the freezer, ship it back to the person, and that vein to vein time is like 2 weeks or longer. So this idea of doing in vivo engineering eliminates all of that. Instead of taking the white blood cells out of the body, you put the engineering vector, the delivery system, directly into the body, and you design it to be specific enough and potent enough that a very, very small amount of it is all you need to engineer enough white blood cells, enough T cells, and then those T cells do what T cells do so well. They expand, right? When they realize that they see some pathogen, now that you've engineered it to see cancer, let's say, it now starts to expand and can recruit other immune cells and really go to work. So, and that's what we're doing with Lucid. So Lucid has a really compelling CAR design. I'll call it a CAR, but let's just say that, they're engineering T cells to do a better job at identifying cancer. And what's so great about what Lucid is doing is they're taking a very novel approach where they're seeing one design that can be used for many different solid tumors, right? They found a common characteristic. And they have clinical data coming out from their ex vivo product right now. And then by working with Syenex, they're able to quickly go and apply that in vivo. So it's almost like a product extension or, a way to get to the next generation of a product they've already been working on for, a decade. And that's what a lot of our partnerships look like right now. People that have, whether it was in academia, we're working on an ex vivo proof of concept initial, design for T cells, or someone that has it in the clinic and is showing success, being able to immediately plug that into a system that then works for in vivo, obviously it saves tons of time, reduces costs, and ultimately allows for patients to get access to these medicines. Because today it's like a fifth of eligible patients in the US actually end up receiving these CAR-T medicines.
I mean, you mentioned Lucid is in clinical trials, right? Talk
Mhm.
to me about how does the regulatory work for platform companies? I'm truly clueless when it comes to this. Like, I know that you can't approve a platform. You guys have to do it via another drug,
Mhm.
correct? So how does that play in with also your philosophy of trying not to, start on one drug and like only do that?
Yeah, it's a great question. What's really inspiring is how much the FDA over the past, I don't know, at least 5 years has been a huge proponent of platform technologies, maybe in theory more than in
Mm.
practice, because there haven't been great examples of this. And all the current regulatory work that's been done has been shaped for drug companies because that's kind of like the state of affairs.
Yeah.
Um, but the idea on the regulatory side is if something's been used before and the only thing that is changing is maybe one piece, then maybe we just need to really evaluate the safety of that one piece, and the rest we can sort of be like, okay, we've evaluated that before. And there's these new designations, advanced manufacturing technologies, platform designation that are still in their early days,
Hmm.
but um, again, it's all sort of this idea of making medicines programmable. And that just means try to abstract away and simplify as many components as possible such that all that's left is like the program you want going into the cell or the modification you want to the cell. I can't say that's all been perfectly figured out yet. So for us, it's learning as we go. We've got a group called Cure Cells that is using one of our ex vivo systems now, entering the clinic. So we've gotten a lot of regulatory experience through that. And they actually have multiple products that they're using the system for. So we've been able to see how, repeatable certain elements can be for a phase 1, even for a phase 2 medicine that they're working on. So there's a lot to be learned. I would say at the center of all of it is by being a platform company, we're going to learn this in a way that no one else will learn it without doing it. You learn by doing. You sort of build solutions to the problems that you choose to focus on. So, but I think there's an opportunity, and I think it's one of the many different things to solve to make it truly as easy and fast and low cost as possible for someone to go from preclinical proof of concept to clinical proof of concept.
Yeah. It's interesting. I feel like the regulatory, the economics are still quite nascent for, in general, platform and tech bio, synthetic bio, this sort of category, this market
Yeah.
of biotech that is emerging in maybe just the last 5 years, right? And maybe you can talk to me about that a little bit and how you and how Syenex is like navigating that. Maybe what has changed in like the last 10 years since you know, did investment banking at Goldman to now when you're running this platform company. And has it gotten easier? Has it gotten better? Maybe what are some things that still need to be improved?
Well, I love seeing all the tech bio companies, and I think a lot of these tech bio companies can be platform companies in the true sense. I still think that there's a bit of a temptation, um, maybe in the boardroom or elsewhere, to prioritize large pharma
Yeah.
to prove that you have built something of merit when there are just so many small drug companies, right, that are all highly innovative, highly incentivized to see a drug work, and they know exactly what problem they want solved. And if you focus on servicing them, you'll focus on a problem that might be commonly held and a solution that is extremely impactful. Um, so I think with tech bio, a lot of companies can be doing that as well. I don't know the stat off the top of my head about how many more emerging biotechs or startups there are today versus 10 years ago, but my gut says that there are more. I think that with genetic medicines, it actually will be very capital efficient to start those companies once you have sort of a disease hypothesis to pursue. And that should lead to more companies. And, the folks at Pillar always really talk about this dream of seeing more drug companies, more founders. I think founder-led biotech is a great example of this becoming even possible. I think a lot of people talk about wanting to see more generalists enter biotech investing. I think
Mm,
biotech has been everyone's favorite category to say, I know my limits, and stop there. But we'll see.
that's fascinating.
Um, so yeah, I think there is a lot of nascency here. There was a time when starting a drug company was super nascent. It was before my time, but in the early 2000s, starting a biotech company, taking it public, assigning a value to a phase 1 asset company was still super tenuous, you know?
Wow.
and that's changed. So
Yeah.
I think it's possible it will also change. I think it's possible that, when we talk about our aspirations at Syenex, people can't map it onto someone else's previous work very easily. And that makes it hard to assign value. And if you can't assign value, it's hard to sort of incentivize other founders to start things. But over time, that can change. And then suddenly people can say, okay, if this is where you're at today, I know what this sort of trend might look like. And that might drive more people to make platform businesses. And I think more platform businesses in biotech will make drug discovery less expensive, lead to more medicines, take money out of the healthcare system, save lives, make people healthier, all these great things.
This sounds so cheesy, but you guys are like genuinely trailblazers in that sense.
In genetic medicines, we're doing something different. It's not proven. It could totally be wrong. It's very possible we look back and,
Yeah.
it was like, oh boy, maybe we shouldn't have done it that way, but it feels right. And as I said earlier, with entrepreneurship, you just need to be passionate about it. And we're willing to take the risk that we're wrong by doing something that feels like it's the right thing to do. And maybe having a little bit of uh, faith in economic faith that, if you create enough value, you're going to see it come around your way. So we know that we're creating the most value this way. We just don't know how it all shake out from here.
Super exciting. I mean, we are here in SF, right, for the JP Morgan Healthcare Conference. And I feel like every single conversation has been about AI X bio, right? How can AI provide value to the entire stack when it comes to drug development, all the way down to automation? Anything that particularly seems promising to you, exciting to you, any particular application of AI you personally are excited to see?
Yeah. I mean, I would say if you zoom out, everything around DNA synthesis and sequencing and our ability to both read and write biology has been a very, very big trend. And that has actually enabled a lot of this bio AI. And um, when you think about AI and you think about sources of data, there's more data in a person than in the entire internet, right? So there's a lot to be done there.
Totally. We need to access all that data.
Exactly. And we are, and we're getting there. And we need to see it all translate into value over time. But in terms of, so I see AI in bio as a continuation of this trend of being able to read and write biology, being able to engineer biology, and now just having even more tools to do that. I love AI applications in protein engineering.
Mhm.
Serge Biswas and Nabla Bio was sort of, pre-Nabla was probably my first real um, exposure to large language models in protein engineering. And this idea that, don't try to hard code in the rules of physics, but just try to let the model figure out the patterns.
Yeah.
I love that. And that seems to have been working very well and has been the fundamental innovation behind lots of these really interesting AI protein engineering companies. And with AI protein engineering, over time, that just dramatically reduces the cost of engineering proteins. So, um, from a Syenex perspective, that's great because we incorporate proteins into these, complex nanoparticles. So now it's cheaper to design one piece or another piece. So, it's like, the cost of steel going down if you're building battleships. So that's good. So AI for proteins is great. AI for cells is more complex,
Yeah.
but pretty fascinating, and should lead to new innovations around how to engineer cells. Cells are pretty complex robots, if you will, and we have no idea how it all really works. Are the neural networks in the cell? How does it work so intelligently? Is a cell a quantum computer? It has a lot of fascinating elements that are hard for us to figure out. So maybe AI in predicting cell behavior and even developmental biology and things of that nature could be uh, open up all new ways of making people healthier. So I like that too. Yeah, there's a lot to be done there, and we're in a very exciting stage in the AI liftoff. Um, And there's a lot of money being invested in infrastructure that is going to be extremely valuable and useful for the next decades. So I'm looking forward to using that infrastructure. And, I'm always happy to see more of it being built.
Totally. I feel like the AI revolution has kind of made platform companies more, I don't wanna say more viable, but definitely more attractive from an investor, standpoint, from the general public. And I think the West Coast is doing quite well there.
Well, I mean, I think a lot of the AI talent is here in San Francisco.
Yeah.
And I think a lot of people like working in the field of bio because uh, the dotted line from what do I do to how do I do something good in this world is a pretty short line.
Yeah.
So that's nice. And the business models are still being figured out.
Yeah.
So we'll see. But it certainly is making it less expensive to engineer biology. And that's a great secular trend. And that's going to lead to better medicines and all sorts of exciting new targets being identified, tools being developed, and the like. So we'll see.
Yeah. I'm personally really excited about PLMs, what they've been able to do for biologics and antibody discovery. As you mentioned, just like letting the model learn biology and then fine-tuning it, or diffusion. Like there's tons of different techniques that researchers are still developing, right? But I think they can make the success rate for these AI developed drugs a lot higher and hopefully reduce costs as well.
Yeah. Yeah. Improving the success rate. That's tricky.
Hmm.
Um, Reducing the cost to then see if something is successful. That's very, very doable. So, shots on goal said a lot, but I think there are more shots on goal. And if it's possible that these models can even help us measure impact faster with fewer patients, that's more shots on goal. So, we'll see. It makes me nervous to imagine the protein engineering models being able to tell you which drug is definitely going to work, but it can definitely help you make some really exciting drugs once you have a therapeutic hypothesis.
Totally. I want to begin to wrap this conversation up just by asking you what you are excited about, whether it's for Syenex or for bio, maybe it's for, generations of talent that you're seeing emerge. Just what excites you right now in the field or your company?
No, the things that I am excited about are I, I'm really there are a lot of things I'm really hopeful about, I'll say.
Yeah.
Um, I'm really excited, hopeful about neurodegeneration
Hmm.
being an area that gets addressed. It's so devastating. It's so common. And there seem to be some uh, threads that span multiple neurodegenerative diseases in the form of sleep disruption,
Hmm.
microglia activation. Your brain's on fire. It's just on fire in different places. So building drugs that can address that will be game changing. I really like longevity, but insofar that it's like what kills you and work backwards,
Yeah.
and make us healthier and stronger. How do I ensure I won't break my hip, when I'm 80? Maybe it means you got to be, jacked when you're 80 and whatever it is, like that's great. And I think obesity medicines have hopefully opened people's eyes to a different way of thinking about human health. Obesity 10 years ago was laughed at as an indication. I hope more people are wondering what's the current obesity? What's the thing people are laughing at today? Um, that they're not taking seriously enough. That is a problem that everybody has, or a lot of people have. I think a lot of the longevity space has these characteristics that could really change the way we think about preventative medicine or just sort of maximizing human health. Those are the ones I'm most excited about. I mean, I think genetic medicines has a lot of promise in all of this. I think we're still in this early days where people will say, well, genetic medicines are expensive, but I really think they will end up being the least expensive
Hmm.
in terms of development. because they're programmable. Yeah. How about you?
I think I've talked through a lot of the things I'm personally excited about is AI, protein language models, all the advancements there for drug development, drug discovery. Um, I I hope that we do end up figuring out, like, what causes neurodegeneration, and we have a more conclusive viewpoint there, because every single article I read is just, like, failure after failure after failure, and it
Mm-hmm.
is, it's for people who work in the space, obviously, and for people who really wanna see solutions to that, it's devastating to read failure after failure. So I really hope we can figure that out in the next 5 to 10 years at least, and start to see some successes there. Your neuroscientist background is kind of coming through, um, but yeah, I mean, other than are you excited about neurotech? Like, it's
Yes.
still very early, yeah.
Yes, I am. I my favorite neurotech idea is a device that helps you sleep faster.
Hmm.
That
There's some companies out there, right?
there are people
Working
working
on
on
that.
going to sleep, but I think sleeping faster is a fun thing to say, and it hopefully begs some questions about what the heck is sleep all about anyway.
Hmm,
Deep sleep helps clean the brain. REM sleep,
yeah.
I don't really know what REM sleep does.
I think it's actually the lightest sleep, right?
It is. So I looked into this before because I know some super sleepers, and when you get to know a super sleeper, you feel this budding envy for their ability to not need as much sleep. And in looking into it a couple times, I think I saw that deep sleep is the same quantity, but it's everything else that's compressed. And then with neurodegeneration, when your deep sleep starts going away, that's when you just fall off the cliff.
Mm,
So deep sleep seems really foundational. Whereas the lighter sleep, I wonder if that's just like evolutionarily, like, you want to be a little bit not comatose in the middle of the night in case something bad happens.
yeah,
So maybe
that's an
that's why it's
interesting
episodic.
thought. I really wanna see something that can help me focus, help me stay awake, actually. Um, I am a caffeine addict, so
Hmm.
maybe a healthier way of, like.
Neurotropics.
Yeah,
Yeah. Maybe that's going to be part of, I mean, who knows, right? Like if part of the way to stay, sharp in old age,
yeah,
and if that really makes a big difference, that can then trickle down.
totally. Yeah, I think there's a lot of, like, potential intersection of these markets and where the funding goes, like, the longevity folks, they can start working on this. I don't know. I don't know if VCs really like that messaging or that market. I'm not personally familiar. I know the public does, and there's a lot of hype around it, but
I think we could get another cortex, prefrontal cortex.
hmm,
That would be pretty cool. With they have all these glasses now, and
yeah,
I think there will be ways to non-invasively interface with people to give them the benefits of an effective additional layer of brain, which could be pretty neat. It's something that I'm still pretty convinced by.
same here. I think for me, automation will be huge. I'm sure you've experienced the grueling hours in the wet lab, um, how long it takes, how much time it takes. So for me, that is one layer of abstraction that if it pans out, I'm super excited to see. But
Yes, 100%. I mean, it is so expensive to start a biotech company because of the way lab work is done and how, every company needs to like buy equipment
yeah,
and do all these things where, where's the AWS of just wet lab?
totally. Yeah,
So maybe Ginkgo will crack that nut. That'd be nice.
hopefully.